Great. Hi, everyone. Welcome to the 11th ActinoBase e-seminar. We have another speaker for you today. This is Satria A Kautsar from Wageningen University in the Netherlands, who's going to be talking to us about his PhD project. So I'll let him take that away now.
Thanks, Emily. Should I share my screen?
Yes, if you could, please.
Okay, so thanks again, ActinoBase, for this great opportunity to present on this very amazing seminars. So I'm Satria. I'm a PhD in the group of Wageningen in Wageningen University, Netherlands, and I would like to have the opportunity to present our effort in trying to map the secondary metabolic diversity or biogenetic diversity of more than 200,000 microbial genomes and metagenome assembled genomes that we collected from public databases. So to perform such a large-scale analysis, we developed a new tool called Big Slice. So it enables really scalable BGC homology analysis tool. Okay. So yeah. For many of us who were working with bacteria, especially actinobacteria, most of us specifically interested in their natural products or the secondary metabolites that they produce. And those natural products are a great source of useful compounds that we can use for antibiotics, antiviral drugs, anti-cancer drugs, etc. So starting from in the 50s, even since the discovery of streptomycin from Streptomyces griseus, which is a dwelling actinobacteria, up to the recently emerging venture on mining natural products from marine microbes. For example, the salinasporomides from the Salinaspora tropica, which were discovered by the duo Bill Fennikel and Paul Jensen in 2003. So it seems that many of us were really fascinated by the natural products produced by these bacteria, not only because they are actually really a great source of novel compounds, but also the fact that when we manage to culture and isolate this producer bacteria, it is much more straightforward to take them and mass produce them using bioreactor, for example, compared to natural products from plants, where most of the time you need to actually grow the native plants and then try to extract the compounds from the large biomass of the plants. So most of us always seems to know in the back of our mind that the current compounds and also the cultured bacteria and fungi, so microbes that we know, we are only scratching the tip of the iceberg of the actual diversity, compound diversity, and also microbial diversity that are out there provided by nature. So things like previously unculturable marine bacteria, which were now started to get really get tracked in the methods to culture them, but also even from the cultured isolates, there are a lot of pathways that cannot be expressed or didn't get expressed in the lab setting. So cryptic pathways, silent pathways, and also not to mention the microbes that are currently hard or impossible to culture with current methods and technology. But really, we know that there is a large iceberg, but can we actually measure how large is the iceberg? So it's important for us to know really what's the capacity really that are out there in nature for novel natural products. So we can, for example, try to measure them from the current compounds that we have isolated, but it will basically only tell us the information about what's actually the tip of the iceberg, how large is the tip of the iceberg, because these are only what we have known. Or also we can try to estimate from the estimated diversity of the microbes species itself, but there are also the fact that not every microbes are the same. So some bacteria, even within species, the strains can produce really different level of novelty in terms of the natural products that they produce. So in this way, we think that genomics can provide a way to map this global biogenic diversity of natural products. So through the analysis and study of biogenic gene clusters, which is the group of genes that are clustered together in the genome, in the microbial genomes that involve together in secondary metabolites. And this enables tools like ActinoBase to mine and predict biogenic pathways encoding secondary metabolites from the genomes of the microbes. And ActinoBase has been widely used ever since its first release almost a decade ago in 2011. And also one way that we can try to chart this map of biogenic diversity is to actually perform networking analysis or clustering analysis of the BGCs. So because those BGCs act as some kind of proxy to the metabolites that they encode, group of BGCs that are similar, we can predict them to produce similar compounds also, so specific chemical moiety. So for example here, 6 years ago, a paper co-authored by also my supervisor Marnix, they tried to collect 10,000 BGCs from 1,000 genomes, bacterial and fungal genomes, and this networking analysis was done to then be able to see this like a group of BGCs that were associated to specific chemical pathways. And recently, last year, in our Big Scape and Corazon paper, we take 74,000 BGCs from 3,000 actinobacterial genomes and then perform similar networking and clustering analysis in order to group the BGCs into families and get an idea of their diversity. However, if we look at the currently available data in public databases of genomes and of BGCs, there are much more data that we can leverage to really get a complete picture of the global scale diversity that are out there. So for example, in ActinoBase database, we have more than 150,000 BGCs from the replicated bacterial genomes in NCBI, and also the Joint Genome Institute have the IMG-IBC database comprising 400,000 BGCs predicted from their collection of 60,000 bacterial genomes in IMG. And also within MyBig, we have a reference for known and experimentally characterized BGCs with known compound products that we can use to basically have a way to connect these putative BGCs together. So in this study, we want to take things even further. So we collect all the bacterial genomes in NCBI in RefSec, and also add some fungal and archaeal genomes. But also we also incorporate some metagenome assembled genomes from several studies. So this will, the idea is to get really like a complete picture of all the not all, but most of the sequenced genomes that are out there and see their biogenic diversity. So this total to more than 1.2 million BGCs predicted using ActinoBase 5 of different classes and also of different lengths from short BGCs, 10-20 kbs to longer ones, 60-70 kbs, even larger than 80 kbs. So you can imagine that the first challenge will be the sheer volume of the data that we need to process. So there is a main bottleneck with all current approaches, including Bigscape, because we rely a lot on the construction of pairwise distance metrics. So if you imagine you have these three BGCs, A, B, and C, what Bigscape does is first we compare between A and B, B and C and A, and come up with like similarity metrics. So in Bigscape, we compare their content of the PFAM domains, but also the synteny of those PFAM domains between the two BGCs. And also for the shared PFAM domain, we compare their sequence similarity. So then we set a threshold from the distances to build a network, so to build a graph where edges represent connectivity, meaning that the BGCs are similar according to the threshold. And using this, we can then group the connected networks, connected nodes together in forming the gene cluster families. So one issue with this approach is that you can imagine that if we want to take, say, 10 BGCs, we will need 10 squared times calculations, meaning that it will scale quadratically. And it will definitely not scale well for more than 1 million BGCs. As you can see here, the runtime is near impossible to finish in our lifetime. So we come up with an alternative approach. So in Big Slice, what we do is that rather than comparing BGCs against another, we represent BGC and plot them into Euclidean space, so Cartesian coordinates. Here, as an example, you see that each BGC have a coordinate in X and Y, so we call this feature. So each column is a feature. And then you can immediately see here that A and B is closer together by I, and without having to actually calculate the distance between A, B, and C, we can try to average the position of A and B, for example, to get a dot. We call them centroids, and then we take a specific radius from that centroid to define the BGCs. To define the gene cluster families. And in Big Slice, rather than 2 dimensions, we actually use 6,882 columns or features or dimensions. So each column, as you can see here, basically represented by a PHMM model that I will tell you later. So there are 2 sets of PHMM models that we generate for Big Slice. One is what we call bisynthetic PFAM. So it's simply a collection of PFAM domains, so from the PFAM database and also some from Antismesh, that we manually filter for things that on domains that are related to bisynthetic enzymes. So for this, we simply calculate their absence presence, so 0 for no hit of the PHMM in a BGC, and 255 or 1 if there is at least 1 hit for that BGC. So similar to Jaggard distance in Bigscape. And the sub-PFAM, so with the pairwise comparison approach, we can compare between sequence similarity of 2 protein sequences within the 2 BGCs. But here we cannot do that anymore. So one way to alleviate that, so that we can still have some sensitivity of the protein sequences, we calculate for each we call core PHMM. So things like catosynthase or A domains, we calculate subfamilies of those protein families. And the way we do that is that we take protein sequences from UniProt, we HMM scan them, and for each aligned sequence for each domain, we perform hierarchical clustering, and then to call the clades, and for each clade, we build PHMM. Here, for example, MP binding subcluster 1, MP binding, sub-PFAM 2. And then we take ranked values according to the bit scores that were represented in each domain. So the best hit will have the full 255, while the second best hit will be lower, and so on. So it will act as like a signature for every domain that were extracted from the BGCs. And using this approach, we managed to process the 1.2 million BGCs in our large data set within 10 days of runtime in our 36 cores CPU server. So this is actually pretty much the time that it took for us for Bigscape to process the same like 74,000 BGCs that we use in our paper. So this is really a remarkable improvement. However, you might be wondering, so this looks nice, the improvement, but what do we lose by doing this basically a heuristic approach on the homology analysis? So what we lose is, one, we will lose some sensitivity, and the second is that we can only rely on a single threshold, meaning that if you remember the picture about the radius, we can only use one radius that we use for every gene cluster families that we want to define. So here you can see the problem appear when we, so the left one, the y-axis is the manually defined groups of some of the MyBig BGCs. And on the x-axis is the gene cluster families predicted by a big slice. So on the blue one here is all the singletons. So this one are curated groups that are, so the threshold were set to string for those groups, meaning that the groups were then be split into singletons in big slice. While on this red one are the groups that are actually multiple groups grouped together into one GCF in big slice. So they actually have a threshold that were set to loose for them or too high for them. However, if we look into the features of the problematic groups, we can actually see by hierarchical clustering that the features extracted by big slice actually able to capture those pattern of each group. Here you can see that the colors representing the manually curated groups are tend to be grouped together. So BGCs within the same curated groups tend to be near each other in the hierarchical cluster. So yeah. So using this approach with this analysis, we can then we did then generate this what basically is a bird eye view or a world map of the global diversity within those 1.2 million BGCs. So we put them into 500 bins. So to collect those BGCs based on their biosynthetic features. And from here, you can, for example, see that in these pink bars, you can see that even though they are only like comprised of around 2,000 BGCs, apparently BGCs in MyBig already done a pretty great job in covering like the biosynthetic diversity that are out there. And also here, which is really apparent here, the purple bars so it seems that the fungal BGCs, especially the NRPS PKSs, are having really like their own characteristics and their own groups separated from those of bacteria and archaea. And also, for example, one other thing that is apparent here is this interesting clade of here is all the rumen, chicken cacum, and human's gut metagenomes. So these are all ribosomal peptide BGCs that really enriched in those like stomach metagenomes. And also for us, what's important thing that we can take from this phylogram is you can see here that the feature occupancy, meaning that the features, the number of that are extracted from a BGC are generally lower in this case, in this region. And usually it also correlates with the BGC length. So larger BGC comprise larger genes and having much more features to extract. But also there are some BGCs that simply we didn't have enough coverage in terms of the PHMMs of these classes, like bacteriocins, etc. So this is one homework for us to be able to, ideally we need this red bar to be like saturated to alleviate this problem. So using this, we can then answer the question that we first asked in our early slide. So here are the iceberg that we present. So each node is a gene cluster family, and this is plotted. So these orange ones are GCFs that we designated as related to MyBig BGC. So they have a close distance to at least one MyBig BGC. And here you can see all the remaining BGCs that have really distant similarity to any MyBig BGCs. And even from there, you can also see that some GCFs contain only metagenome BGCs, meaning they are exclusive to the metagenomes. However, some words of caution. So these numbers should not be taken as face value, because like every clustering, it should always be an exploratory effort. So there will never be like a universal right or wrong. So usually what we do is that we test multiple parameters, multiple algorithms, and then we interpret each of the results and with each of their reasons. So one last thing that we did for the analysis of this result is we try to see which taxa or which species have the largest potential for novel singular metabolites. So here, for example, with very strict threshold of 300, you can see many actually gram-negative bacteria aside from the Streptomyces and some fungi. So this probably due to their large numbers of the genomes in the ISBA. But actually, if you take the results from the more looser cutoff, meaning that the diversity will be like more generic and more abstract, those many of those gram-negative bacteria are actually drop down in ranks. So this suggests that the diversity of the BGCs from these bacteria are bound to like single or multiple genes, so deletion or insertion of genes rather than a recruitment of an actual novel BGCs or group of genes. So here you can see that all the species having really high unique GCF counts. So many of them are actually actinobacteria, especially Streptomyces. But also there is this one node which is Fusarium oxysporum, a fungi. So we haven't really taken a look deeper into interpreting these results, but yeah, feel free to discuss or give your thoughts in the Q&A session. So yeah, other than this analysis, Big Slice also provide this nice visualization for daily users. So if you run a Big Slice clustering, then you have this you can run this visualization engine to explore your results and your data sets. And using that engine and also the results that we have, we also is currently building this database of gene cluster families where you can take anti-smash results. So you can copy paste your job ID and then you can put that into BigFam database, and then immediately get all the queries of all the gene cluster families that are closest to that BGC. So this enables you to quickly see what are the BGCs related to this BGC of yours, and also how they are distributed within the taxonomy. Oh yeah, one last thing is that apart from the large-scale clustering analysis and the visualization, one hidden gem of BigSlice is that we store everything in a SQL-based data storage. Sorry, I think it took a bit of a while. So what this enables you is that you can then perform a very complex query. Here, what I do is that I query for all protein sequences from Streptomyces that have a hit for a domain. So here you can see that it performs it really fast across those 1.2 million BGCs, and we can then use this in combination of scripts and also of Python softwares to actually perform all kinds of analysis. So here, for example, I use this to try to plot the distribution of those sub-BFAM domains of a domain and compare them between the data sets. And also all the analysis that were done before, and also the software visualization was also done upon this SQL-based data storage. So in the end, there are still more works to be done, a lot of works. So we greatly appreciate your feedback and contribution in improving BigSlice and BigFam. But also the analysis here that I present here is really just a preliminary analysis, and we plan to really get into details on that with Nadine Zimmer from the University of Tübingen, and also her PhD student Athena, to really try to look deeper and perform more robust analysis. And also we plan to actually look for a way to make this even more catering your bench biologist daily needs. So I talk about this with Emzo from Warwick, who are working on these cluster tools that enables quick queries of BGCs or from genomes based on their BFAM domains. And also we plan to also see if we can use BigSlice platform to extend beyond the biotic domain. So for example, to see the distribution of antibiotic resistant genes that can help us prioritize the BGC. So we plan to do this with Mohammed, a previous PhD student from Nadine, who are now working with us as a postdoc in the Metabolic group. So yeah, thanks a lot. Sorry for taking too much of your time, and yeah, really happy to take your questions and discuss. Thank you. Should I stop sharing?
Sorry. Very much, Satria, for that fantastic talk and what fantastic goals. Just wait and see if anyone has any questions via Twitter or on YouTube.
Just check. Yeah. Yeah, I think we have a question from Professor Rifon. I checked the YouTube chat.
Right, sorry.
Yeah, so there are questions from Professor Rifon. Did you also find some known potent producer in your list of rich strains? Not yet. So as I mentioned, we haven't really done any further deeper analysis on the results that we have now. So yeah, apart from the list of like a mix of those seemingly pathogenic or symbiotic bacteria and fungi, and also some streptomyces who are already known as like the potent producer of them, we also see some Burkholderia, which were now really like getting tracked over and over again. But yeah, we haven't really looked at those deepers, and also, yeah.
Another question from Enzo de los Santos. He says, great talk. Did you look at any of the faraway clusters from Streptomyces or some of the other species? How different do they look?
Faraway clusters, so meaning that BGCs that are not exist in Streptomyces, I guess. So yeah, we haven't really looked at that, but from some basically some like cursory look that I took at the preliminary results that we have, there are indeed like GCFs that are not contained within the actinobacteria or within the Streptomyces. But what we see is really like the diversity is really skewed towards the actinobacteria and the Streptomyces, like the almost like 1/3 of the like novel BGCs are in the Streptomyces, and the others are like shared between the other species. Yeah.
Right. Okay. And Riza Pusranto asks, he says, great presentation. Could you explain if your analysis would be better using UniProt, or could you also use some other databases?
So this means the Sapifam, I guess. So yeah, actually, there is, well, honestly, there is really no specific reason why we choose UniProt. So for the Sapifam generation, what we basically need is a way to actually have like PHMMs representing different kinds of protein sequence that can happen in each protein family. So, for example, all the possible combination of A domains that can be out there in nature. So I suppose that we still find that some core domains, like the one that encode for bacteriocins, for siderophores, they really have really low representation in UniProt. And it will be nice if we can have data, more proteins on that family, so that we can build more like larger Sapifams out of there to improve the features. Yeah.
Scott Yarmish asks, will there eventually be a way to view your hypothetical new bacterial genome against the global analysis you've done? And any way to score or rank a new genome to those looked into?
Yeah, definitely. Actually, this already like in my mind. So like for my next basically journey, so for my like developing a platform to enable really like the management of sequence genomes, like from the raw rates, etc., up to the like automated analysis of the novelty of the BGCs within the genomes, etc. So yeah, definitely.
Justin Vanderhoof says, great. You indicate that MI big gene clusters are distributed well across the global map, but isn't that also because as we slowly tap into the unknown biodiversity?
Sure. Yeah, definitely. Like that, there is also so that's a very good point. So because what we use for building the databases for our current analysis is what we have from AntiSMES, right? So meaning that we only capture like well-defined BGCs that we know derive from all the BGCs that we have characterized. Basically, the way AntiSMES rules work is that usually if you have known BGCs producing known classes, then you develop rules for that type of biosynthesis. So meaning that we only capture things that we already know in some sort of way. So there is also a possibility for us to yeah, so for example, we can try to define more clusters and then really look at their core biogenetic domains that represent those clusters, and then try to explore more. But yeah, the platform really could enable that. I think it's pretty simple to do. Yeah.
Jonathan Paravilibos asks, he says, thanks, Satria, for a great talk. Did you find any differences between the metagenome assembled genome and the regular sequence genomes?
You mean like difference like within the content of the BGCs? Yeah, as you can see from some of the results that I showed before, like there are some gene cluster families that only can be found in the metagenome assembled genomes, and yeah, those basically represent things that. So we have only 20,000 metagenome assembled genomes compared to the almost 180,000 of those from NCBI RefSeq, which we assume at least they define that those are isolated genomes that come from cultured bacteria. So yeah, if we also try to incorporate more metagenome assembled genomes, and also even like metagenome context from metagenome sequencing data, we can then get better complete picture. Yeah.
Right. I think that's all we have time for today, but thank you very much for the talk. And I'm sure people can reach you on Twitter if they have any more.
Yeah.
And we'll be back on the 6th of August with another speaker. So we'll see you all then. Thanks very much, everyone.